Please be seated. I open this public session of the Doctoral Examination Committee of the Faculty of Medicine from the Christian origin of Vrije Amsterdam to the polygrammatic academic community that we are today. Our scientific practice is intertwined with fundamental values and a sense of wonder. Prior to the examination, I invite the candidates to give a brief explanation of the research on which the thesis is based. The floor is yours.
Dear Rector, dear guests, from the year 1990 to 2016, the number of Parkinson's patients worldwide had more than doubled, with an estimation of more than 6 million patients living with Parkinson's. This would have likely come as a shock to James Parkinson, who more than 200 years ago discovered and described the disease, calling it the shaking palsy. Based on only 6 patients, some of whom he just barely saw in the street, he described it as involuntary tremulous motion with a propensity to bend the trunk forward and to pass from a walking to a running pace, the senses and the intellects being uninjured. But while motor symptoms in Parkinson's disease can be so striking, they truly represent only the tip of the iceberg, because Parkinson's patients suffer from many more symptoms that are non-motor. From constipation to speech problems, swallowing difficulties, sleep difficulties, depression, anxiety, hallucinations, and even dementia. So much so that in the past years, we've come to redefine Parkinson's disease as one where the intellect is heavily injured. And if you take Parkinson's motor symptoms, add to them memory loss as in Alzheimer's disease, and other symptoms such as visual hallucinations and particularly dementia, you end up with another disease, dementia with Lewy bodies. Dementia with Lewy body is basically the second most common cause of dementia worldwide. But it's not just a combination of Parkinson's and Alzheimer's disease, because dementia with Lewy body patients progress so much faster and they survive much shorter. So if it is fair to say that dementia actually kills, then it becomes crucial to solve the mystery of cognitive impairment. Is it possible to look at the brain and ask the question, where in the brain does cognitive impairment occur? And so to answer this question, I would like to tell you the story of the insular cortex, which has for the past few decades has been the mysterious island of the brain. There are several reasons why we would need to study the insula in neurodegenerative diseases, because of its very special features. Number one, it has very specialized neurons, phonoeconomal neurons, which function different. They look different from the rest of the neurons in the brain, and they're presumed to play a very important function, self-awareness. But also, if we look at the insula as one region, it truly is not just one region. It's a region that is subdivided into several other regions. And why this is important is because each of its subdivisions actually has a very different architecture. And so each subdivision has its own architecture of layers and own architecture of neurons. Even the types of neurons are different. What this would translate into is that each part of the insula would also have a different connectivity profile to the rest of the brain. And then each part of the insula would have altogether a different function. So that if we look at the functions of the insula, we would see that the insula is actually performing so many functions together, from subjective feelings to autonomic functions such as the heart rhythm and the blood pressure, to even feeling the internal state of your body, like just the fact that you feel your heart beating, to cognition and decision-making and emotional symptoms, and so many others. And so in order to be able to study the insula in neurodegeneration, we would have to look at it from different angles. One, by zooming in at it at the micrometer level, looking at its cells and the pathology that's in it, and then zooming back out, looking at it as a station within a network. And this is what I would like to start with. In this thesis, I started by looking at the cells within each part of the insula, and then the fibers. These fibers are very important because they allow communication of neurons with each other, and they allow communication of the insula with the rest of the brain. And we even looked at astrocytes, which are seen here in red, these cells are important in terms of supporting neurons. And next we ask the question, what kind of pathologies exist in the insula in Parkinson's and dementia with Lewy body? Is it only Lewy bodies, pathologies we normally see in Parkinson's, or do we also see a bit more, such as Alzheimer's pathology? And the results were actually striking. One of the first results we had was that the phonoeconomal neurons that are presumed to play a role in self-awareness were actually affected in Parkinson's dementia and dementia with Lewy bodies. And we also saw that fibers allowing communication between neurons were severely damaged, and more so in dementia with Lewy bodies compared to Parkinson's. Even astrocytes, which you can see here in red and connected to so many neurons, these cells were damaged, and they even had pathological aggregates. And in terms of pathology, we saw not only Lewy bodies and Lewy neurites, which we normally see in Parkinson's, but we also saw pathology in the blood vessels, plaques and tangles as we see in Alzheimer's disease and other pathologies, bearing in mind that each pathology has its own method of injury in the brain. And so the more the pathology, the worse off the patient. And then we needed to zoom out and look at the insula from the perspective of a network, looking at the brain as basically a network with different parts of the brain as central stations connected to each other, so that in the end they can perform the complex function that the brain does. What we first found was that the functional connections of the insula were disrupted in Parkinson's disease, but in a very peculiar way. The networks that altogether work with the insula to control cognition were affected, and the dynamics between them was also affected. And this had a correlation with cognitive impairment in Parkinson's patients. And then we looked at what particular structural connections between the insula and the rest of the regions in the brain would be affected in Parkinson's and associated with cognitive impairment. And we found one particular connection that was damaged, the connection with the cingulate cortex, a region in the brain that looks very much like the insular cortex. It has the same subdivision, it has the same function, and even has the same phonoeconomal neurons. Together, they work together to perform very important function related to cognition. And this particular connectivity was affected in Parkinson's and associated with cognitive impairment. But it didn't really stop here. We wanted to know the implications of the insular damage on other neurodegenerative diseases. So we incorporated data from frontotemporal dementia and Alzheimer's disease, and we saw that the insula was damaged in several neurodegenerative diseases, and that the resulting deficits of that damage were huge, not just cognitive impairment, but also emotional deficits, behavioral problems, speech problems, delusions, hallucinations, all kinds of deficits resulting from this damage. So that all together in this thesis, we could put every piece together, look at the different parts of the insula, the different connections, the cells, the astrocytes, and the accumulating pathologies. And we could point at the insula and say that this region is in fact the vulnerable island of the brain, a hub for pathology, degeneration, and a major contributor to cognitive impairment. Thank you.
Thank you very much. May I hear now if you're prepared to defend the thesis?
Rector, on your authority and as authorized by the College of Deans and in accordance with the Doctoral Examination Committee in the Faculty of Medicine, I am prepared to defend my thesis entitled The Vulnerable Island, Pathology and Connectivity of the Insular Cortex in Parkinson's Disease and Dementia with Lewy Bodies, in order to obtain the degree of Doctor of Philosophy.
Thank you very much. And the opposition will be kicked off by Professor Parkinson. She is the Professor of Translational Pathology at the Department of Clinical Neurosciences of the University of Oxford in the UK. Very warm welcome to you, traveling all the way from the UK to here, and the floor is yours.
Thank you. On the authority of the rector and by virtue of my right, I would like to ask you some questions. But first of all, I would like to congratulate you and your supervisors for your achievements today. If we start with the most obvious questions that we have really, what has really brought us over here today, what makes this island of insular cortex so selectively vulnerable for neurodegeneration? And you can be relatively brief because we will return to this issue later.
Yeah, it's learned upon, and this is exactly what we try to understand. When we first studied the work on the insula, there was no evidence, much evidence from the neuropathological perspective, but there was a lot of information from the imaging perspective showing that the insula had some receptor binding deficits early on in Parkinson's with mild cognitive impairment. And also some studies had pinpointed that the insula is affected in over 20 neurological diseases. One of the main possibilities of why it could be vulnerable is because it's presumed to function as a hub, and hubs are very active. They integrate lots of functions, and therefore they have a lot of needs for blood flow, and they are more susceptible to degeneration.
And what is your understanding of how is Parkinson's disease a good disease to study selective vulnerability, which obviously involves all of the neurodegenerative diseases?
Yeah, I believe it does.
Because of the
Yeah.
neurodegeneration being very specific to certain regions.
Yes, so because we know from the Braak staging how the spread of pathology goes, and then we know that certain regions are implicated at certain time points in the disease, and the insula is affected in Braak stages 5 and 6 of the disease, so a bit later on, but it is implicated as one of the regions that is particularly affected, yeah.
So you have studied specifically alpha-synuclein, tau, and A-beta pathology in the different subdivisions of insular cortex. Would you mind just briefly summarizing those results and regarding these three pathologies and regarding the three disease subgroups that you have examined?
Yeah. So we first started by looking at whether alpha-synuclein has particular distribution throughout the three different subregions of the insula. And we found that it had a decreasing gradient from anterior to posterior, so that you had most pathology anterior, and the more posterior you go, you had less pathology. So then we went on to study phosphotau and beta amyloid. And we saw that phosphorylated tau had a very similar distribution pattern in the insula, which is basically much more in the anterior insula and then has a decreasing gradient towards the posterior insula. But beta amyloid had an opposite distribution. It was most severe in the posterior parts and then would become less severe in the anterior parts of the insula.
So the alpha-synuclein and tau is higher in the agranular versus the disgranular layer. But if in page 94 you look at the kind of the graph that you've given, I realized that you do have a couple of, or actually one outlier on the agranular group regarding the synucleinopathies. And I was wondering if you would have removed that one outlier, whether the significance would have still existed.
With regards to alpha-synuclein or to? Regarding
the alpha-synuclein, yes, on page 94.
Yes,
so the top right.
I believe it would still exist. We also studied it in another, in similar cohort in the previous study, and we still saw the same result.
Okay. What is there a theory that you could kind of explain why the tau and the synuclein seem to be affecting another subregion than the beta amyloid?
Well, it seems to follow the overall distribution pattern. So for beta amyloid, the tau phases begin with the cortex and then go down. And for the insula, it's the same. For the insula, we see more beta amyloid in the posterior isocortical part of the insula, and then much less in the limbic and the transitional part. And the same goes for phosphorylated tau and alpha-synuclein with regards to their Braak staging and distribution. The same happens in the insula, so it's exactly the same thing with phosphorylated tau and alpha-synuclein starting more limbic and then spreading to cortical. That also happens within the insula.
Okay. And what about what kind of immediate impact do you think that these findings really have for current practices?
From a clinical perspective,
or from a neuropathological assessment point of view?
Yeah, from a neuropathological assessment point of view, it makes a difference where you're looking at in the insula. From a diagnostics perspective, most of the time we're looking at one particular part of the insula to provide the diagnostics, whether for phosphorylated tau or alpha-synuclein or other pathologies. But it really does matter whether you're looking at the ventral part of the anterior insula or the dorsal part or the posterior insula, because the distribution across them is not the same. And then these regions are susceptible and vulnerable to different pathologies. So the anterior part is more vulnerable to alpha-synuclein and phosphorylated tau, but the posterior part is more vulnerable to beta amyloid pathology.
But the current staging criteria, for example, the Parkinson staging criteria, does that include examining the insula?
I believe it does.
Not at the moment, but I think after your thesis, it very much should too. What about one proteinopathy that you have not examined, TDP-43? Was that a conscious decision not to include that? Is that just that there was not enough data?
Yeah, so initially, well, we started with alpha-synuclein. It's the main protein in Parkinson's. And then we wanted to see other pathologies, but we didn't have a particular a priori hypothesis on what TDP-43, what its distribution would be, or particularly what we would be looking for. We wanted to look at the most common pathologies, beta amyloid and phosphorylated tau and alpha-synuclein. And then by chance, we also saw our gyrophilic grains also within the insula. So we didn't have a particular hypothesis for TDP-43.
Okay, but you mentioned RTAG. Would you explain to the audience what does that mean?
Yeah, so it's age-related tauopathy, astroglial tauopathy, and these are astrocytes that do have tau aggregates within them. In this thesis, we did not particularly study them, but we found that they were very prominent in most of the cases. Most of the cases had astrocytes with tau pathology in them.
This is the study where you are trying to examine the influence of concomitant Alzheimer's disease pathology.
Yeah.
Do you think that the presence of RTAG could have skewed your results? Because in your patient samples, 9 out of 10 and 3 out of the 5 DLB cases did have the RTAG.
Yes, I believe it depends what result, because we see extensive pathology. We see everything in the insula, particularly in DLB. There's also astrocytic synucleinopathy. So the insula is actually just swamped with all kinds of pathologies. So I wouldn't be able to pinpoint RTAG and say this would skew my results, because there's actually a lot more pathological aggregates in there.
I see. But what was the inclusion criteria for you selecting the cases for this study?
Yeah, the inclusion criteria were cases that fit the diagnosis of Parkinson's clinically, and that neuropathologically as well, and that the insula itself had no deficits. So there was no atrophy in the insula, there were no other damages in the insula, and that patients did not have a long history of neuropsychiatric diseases that could confound our cohort.
Okay. Well, thank you very much.
Thank you, Professor Parkkinen. From Oxford, we moved to Groningen, which seems a long trip, but we are virtually joined by Professor Van Laar. He is a neurologist and affiliated with the Department of Neurology in the University Medical Center Groningen. Professor Van Laar, welcome, and the floor is yours.
Thank you, Mr. Director. On your authority and by the virtue of my right, I really like to ask some questions, and I would like to start with congratulating you and your team with this beautiful topic. Very few people pay such a huge attention to this vulnerable island, and you did very well, but I still have some questions, so it's a nice basis to talk about that. I would like to start with chapter 3. And in chapter 3, you describe very nicely this gradient of at least the alpha-synuclein pathology from a more anterior to a more posterior, lower percentage of cells being influenced by this alpha-synuclein aggregation. So That sounds nice that there is a gradient. And the first question I would like to start with is, do you understand why this gradient might be there? Is there any reason to think there should be a gradient, or were you more or less, let's say, were you astonished by this finding?
Learned opponent, thank you. So indeed, we did not know, because there's very scarce evidence on pathological aggregates within the insula. And there was only one study that described phosphorylated tau being more prominent in the anterior insula than as opposed to the posterior insula, but there wasn't any other information. But I do believe that this gradient makes sense, because in the distribution of phosphorylated tau and alpha-synuclein, not all regions are affected at the same time, and not all regions are affected in the same way. And so there is a general distribution pattern for all the different proteins and all different pathologies. And that is what Braque is describing in his different stages. And the insula just really represents that in one region, because it's one region that's divided into three. And each of these regions represent different parts of the brain. And the way that is pathology is distributed in them represents the way it's distributed in the rest of the brain. So I believe in the end that this gradient makes sense.
But that's quite a broad remark. What do you mean exactly by this represents the alpha-synuclein spreading in the brain? Because as far as we know, I think in the literature supports, it's a kind of synaptic transmission which follows a certain pattern. But why is this pattern, and how does this relate to this pattern in the rest of the brain?
Yeah, so for example, alpha-synuclein starts off in the brainstem, in the olfactory, in the vagus, and then goes on to the locus coeruleus, and then the midbrain, and then goes on to the mesocortex, and then finally in the cortex.
Yeah. Precisely
Why it has that particular distribution, I believe we don't yet have an answer to that. And because the same for phosphorus.
The reason I ask it is because I'm a little bit doubting this is really the case. And I'm not doubting on your data, but if I look at your methodology section, you described nicely how you got the brains. And the most striking thing was, at least for the PD and PDD and DLB brains, it has a huge variability in duration of disease. So you examined brains in between 2 and 26 years of duration. So what does it mean to conclude there is a gradient in such a huge variety of duration of disease? Because if there is a spreading, and I completely agree with you, there is a spreading from one place to another, and you described some of the processes, and very likely there are subtypes, as you know, but perhaps on that later. But what does it mean if you just examine cross-sectional brains of 2 to 26 years and then lump together everything and say there is a gradient? What does it tell us?
Yes, I completely understand the difference between them, but from a neuropathological perspective, we look more at the Braque staging. What Braque stages do these particular brains fit within? Are they Braque stage 4, 5, and 6? So, and I believe there isn't quite a proportional relationship between disease duration and Braque staging, that there's probably not a very precise timeline for when you have pathology that you should progress to Braque stage 6 within, for example, 4 or 5 years. So I think this relationship is not a very proportional relationship. So in order to argue, so I cannot argue that the patients of 20 years would be particularly so very much different from patients that had a disease duration of 5 years, except by looking at their Braque staging. What Braque stages were they at, and then looking at where in the insula is the insula affected, yeah.
Okay, but do you think this might have influenced your conclusion? Because you said, okay, Braque stages, but that would implicate that there is a huge variability between, and I think there is a huge variability in the speed of the process of spreading the alpha-synuclein throughout the brain. But I think it's a bit difficult then to say there is a gradient, because this gradient may be in some patients perhaps after 2 years, apparently, because what was the variation actually in the data you found? Because that tells a little bit about the validity of this finding.
So we had a small cohort first of all. So maybe if we are to increase our cohort and our sample size, we would see some differences. But even if we look at incidental Lewy body cases and then dementia with Lewy body, so they kind of represent an extremes on the spectrum. You would see, for example, dementia with Lewy body with a disease duration of 2 years. We had cases with, I believe, 2 or 3 years disease duration and extensive pathology that still was different and still showed a bit of a gradient throughout the insula. And so I do believe that this gradient does represent the vulnerability of different regions to different pathological aggregates.
This type of arguments really help, yeah. Last question, because then I think my time is up. I saw you were very interested in women's health. And that's a very good new topic, of course, since recent years. And we know that these patterns of degeneration are very gender specific. Did you look at any gender specific differences in your brains between men and women? Because, for instance, we have now some cholinergic data that there is a completely different degeneration pattern of the cholinergic system between men and women. And so what did you find in terms of did you look at it? That's the first thing. And can you comment anything on that difference?
Yes, we didn't look at it in the first study, but we did in the axonal degeneration study. After we performed our analyses, we also stratified them based on gender and looked at the association between, with pathology in males and then in females. And we saw some variations between these genders.
Okay, some variation between these genders. Can you be a little bit more specific, or is that too much to know, Mr. Reck?
Thank you very much. Professor Lara, I have to interrupt you on these important gender differences. We'll talk about it later, perhaps at another occasion. Thank you very much for joining us virtually. The next opponent is Professor Hall, and she is associated with the University Medical Center Utrecht, to be specific, the Utrecht Brain Center. Welcome to you, and the floor is yours.
Thank you. On the authority of the rector and by virtue of my right, I would like to discuss the content of your thesis with you, dear candidate. But first, I would like to congratulate you with this very nice book, which I read with great interest, which has beautiful pictures with neuropathology. It's really, really nice. And I also would like to congratulate your supervisory team. It might not be a big surprise that I will talk about astrocytes. I know everybody always expects me to do, and it's also the cell type that I like. And also you mentioned it as well in your talk, and you come back to astrocyte pathology in 2 of your chapters. One of the things that I wanted to ask you, because you talk about these different subregions in the insula. And then you also look at the astrocytes in these areas. Are there differences between the astrocytes, between the different regions?
Learned upon, and thank you. First of all, we did indeed kind of qualitatively look at astrocytes in the insula and their involvement in terms of alpha-synuclein pathology. We did not quantitatively look at that, but we did see that particularly the astrocytes in the anterior insula would show much more pathology. So we, particularly in the agranular insula, we saw a lot of these star-shaped alpha-synuclein looking structures. And when we stained for them, these were just mostly astrocytic alpha-synuclein. But we also saw astrocytes that had along their fibers varicosities. And these seem to be mostly in the anterior part, particularly in the agranular part of the insula. And we tried to dig in as to what they really represent and whether they could be the varicose projection astrocytes, which were recently present and discussed in the literature. But
Yes.
we believe that there's something different in terms of astrocytes in the anterior part of the insula compared to the posterior part.
Okay, so there are probably different subpopulations.
Yes.
I think it's really nice that you discussed this varicose projection astrocytes, which I have to correct you here, are not recently being described, because I think in the 90s, Colombo from Argentina, he already has shown very nice pictures of these astrocytes with these long projections. So it's not really recent, but there's not so much attention for them. One of my questions is, because you also say, because you're now starting on this topic, that we need to study these type of astrocytes more. Do you have any advice for researchers how to do that? Because they are human specific, right? Or at least
Yes.
primate specific.
Yes, indeed. These seem to be primate specific in humans and in apes. And they also seem to be sub-region specific. So I would believe.
So if I want to know what these cells are doing, what do I need to do?
Yeah, so first I would look for them, I would stain for them, and then maybe study them from a more molecular perspective, or just see. So, for example, we did not see if there were any differences if there were any losses in these particular astrocytes within the insula, and whether they, as opposed to other types of astrocytes, would be more vulnerable to opsyonuclein and tau. So I think there's a lot that can still be studied with regards to.
How would you then study whether they are more vulnerable for pathology than the other astrocytes? So what kind of experiment would you do, or staining?
I would well, just GFAP. So they were GFAP positive. So I would.
But is that sufficient? I'm making it a bit more difficult for you. Right? Is that?
No, perhaps there would be other markers that I may not be aware of, but maybe other markers that would be more specific to these particular types of astrocytes would allow us to distinguish between this phenotype and another phenotype. Although I do see in the literature that there are discussions that these may not be per se specific astrocytic phenotypes, but they are responses, astrocytic responses to a particular change in the environment. So there was a study that said these astrocytes seem to be present in diseases as well. And so there's a possibility that it's not just a phenotype, but actually a response of the astrocytes where they just acquire these varicosities. So
okay, so now you're saying these are really related to degeneration.
Well, I understand that these varicosities also play a role in protein degradation pathways.
Okay, that's very interesting. Now we're talking about mechanisms. How does alpha-synuclein end up in astrocytes?
Through endolysosomal vesicles, also probably through nanotube tunnels, and then they're uptaken.
So it's not made by the astrocytes, right? That's what you say. It's taken up, but at what site you think of the neuron?
Where are they taken up from the neuron?
Because I always understood it's a synaptic protein. So how would that work, you think, if you think about how astrocytes interact with synapses?
Yeah, perhaps at the synaptic left, there is an uptake of these. And
are astrocytes always at the synaptic left?
I believe they are.
Tripartite synapse, does that?
Yeah.
So? No, so they are very close to the synapses and can potentially, I don't know whether that would work out, but could take it up. I think the rector is looking at me.
I'm just wondering about the astrocytes. But
oh, okay. so I still have time to ask a question.
1 minute actually.
1 minute, okay.
Yeah.
Now I forgot what to ask. Astrocytes, yes, taking up tripartite synapse. Yeah. So I remember. So if we go to what is it, page 88, there you show with stainings, and you say that alpha-synuclein is actually inside the astrocyte. And if you go to, it was page 88, for instance, and we go to figure Q, then you see these green dots, which is alpha-synuclein, I think, in this case, and the red astrocytes. So is this really the proof? Could that be other cell types? What would the microglia do? Because you do not talk about this cell type at all.
Yeah, we did indeed stain for microglia, and we stained for microglia with alpha-synuclein, and we did not see any stainings.
Okay.
So we could for sure exclude that these are
But do
microglia.
you agree with me that, for instance, picture Q does not prove that it's inside astrocytes?
Yeah, indeed they were. So I think what I mean by that is mostly if you look at also O at figure O and at figure R, and then there are also other figures where we could see that it's very close to the cell body.
Yeah, but it
And
could be potentially in other cell types, I think, still.
yes, of course. Yeah.
Thank you very much. I'll give the word back to the rector.
Thank you very much, Professor Hall, for this astrocytic cross-examination. Professor van der Werf is the next one. He is a chair and also affiliated at the Department of Anatomy and Neurosciences here at the Amsterdam University Medical Center. Welcome.
Thank you very much, Mr. Rector. On your authority and by virtue of my right, I would also like to take this moment to involve the candidate in a discussion about the work presented here, but of course I will not do so before having engaged in a few words of praise, because I think we're looking at a very nice and very attractive thesis that we see before us. Your favorite structure, the insula, somehow is represented by a black hole in the middle of the image, which presumably represents the lack of knowledge that we have about the insula. At least that is my take. It's very readable, especially already in the first introductory chapter. You lay down the anatomy of the insula and the functional anatomy in a very scholarly way, and I found it very, very enlightening to read. So thank you very much for the thesis. And I guess we all have our pet structures in the brain. Some people like astrocytes. I know that your promoter spent 4 years of his life thinking about little else than interlaminar nuclei of the thalamus. And I think you and I share, in a way, an interest in these slightly mysterious areas of the brain, like the insula, truly remarkable area. With so many interesting findings. So let's cut down to the chase and talk about your findings. We've heard about this gradient already with the bulk and the brunt of the atrophy, both in MRI and in neuropathology, seem to center on the anterior insula. In a way, I find that not as remarkable as the inverse, namely that the posterior insula is relatively spared. So what constitutes the sort of the resilience of the posterior insula? And I give you a few considerations. One is that the posterior insula is neocortical as opposed to the anterior. And as we know, neocortical seems to associate with neocortical. We know that it, and you describe this, it connects to the prefrontal, to the parietal, to the somatosensory, areas that in the cortical domain are early affected in Parkinson's. Why not the posterior insula? Let's start there.
I learned upon, and thank you for your words. Indeed, we do see from both pathology and connectivity studies that the posterior insula is not as affected as the anterior part of the insula. There may, as you have mentioned, that it may have to do with the fact that it's a neocortical, and so it has the layers. For example, one of the differences is that the posterior insula has a very dense layer 4, layer 2, which you do not find, which you do not find in the anterior part of the insula. And another thing is that the myelination starts from cortex and then goes down towards more allocortical regions. And this timing of myelination actually confers some more protection to the myelinated fibers in the neocortex as opposed to the allocortical part of the brain. And so there may already be some features in the posterior part of the insula, inherent features in it, but also the fact that pathology does not reach the cortex until very late in the disease.
True. So you ascribe it in a way to the internal intrinsic properties of the posterior insula, and yet I would like to go back to something that we heard from our colleague Des van Laar, who said that we think about the Parkinson's disease process as almost being an infectious kind of process that travels synaptically from cell to cell. And can we understand the sparing of the posterior insula on the basis of the neuroanatomical connectivity? Because I would expect the opposite in a way. I would expect on the basis of connectivity with other regions that perhaps the posterior insula would be an early target.
Yeah, so from, let's say, for example, from the perspective of alpha-synuclein, it would be more in the allocortical regions and then later on to the neocortical parts of the brain. And so from that perspective, you would still expect, even if the spread was synaptic, you would still expect that the cortical part would be affected later on. I believe the same would happen for phosphorylated tau, and probably the opposite would happen if you were to look at beta amyloid. But for example, the anterior part of the insula is affected by phosphorylated tau and by alpha-synuclein almost at similar stages. So Bragg stage 4 for phosphorylated tau, Bragg stage 5 for alpha-synuclein. And these two pathologies seem to also kind of induce each other, their aggregation, and they may have their own damaging effects in the brain. So on that basis, I would still, and the fact that they affect the anterior part of the insula, the allocortical part, much earlier than even from a synaptic perspective for the spread of pathology, still the anterior part of the insula would be affected compared to the posterior.
You're very convincing, so I'll take your point of view, and let's then, as you say, turn to the anterior insula, where we find these mysterious neurons, those von Economo neurons that have spoken to the imagination of neuroanatomists for years and years. Supposedly uniquely human neurons that appear only in a very limited number of areas in the brain that you have investigated. Can you tell us something about the sort of connectivity of those neurons and why they should constitute a vulnerability?
Yeah, so von Economo neurons are present in mammal species, and they were particularly of interest because they were affected in frontotemporal dementia. And they're present in the insular cortex, but also in the cingulate cortex. Much of what's happening now is pretty much assumptions as to what they're performing and how they're affected, but they look different and they have different dendritic morphology. And prior to this, we didn't know if they were also affected in Parkinson's and dementia with Lewy body, but we saw that they were indeed affected. So yeah, why would they be vulnerable? Yeah, I think this is still very difficult to answer because we lack a lot of information on precisely what they do, but they have a monoaminergic aspect to them. And yeah, and we would need to understand what they're doing in Parkinson's and dementia with Lewy body and what makes them vulnerable or not.
Right. I'm looking at the rector to see if he would allow me a small question. Thank you, Mr. Rector. I'm wondering if you can speak to the intrinsic connectivity of the insula. It seems as if you describe the insula as being connected with other areas, but we also know that inside the insula, there is very strong connectivity. I'm going to challenge you and say that probably the intrinsic connectivity of the insula is stronger than that of the extrinsic connectivity. Wouldn't that then still mean that we have a homogeneous atrophy within the insula?
So the insula is indeed heavily interconnected and then also heavily connected to the rest of the brain. And so each part of the insula has its own specialized functions, but so it has its own unique functions, but they all share in some way. So for example, we see that the dorsal anterior insula is implicated not only in cognitive functions, but also functions that have an emotional cognitive aspect to it. So for example, a function that has cognition and emotion together. And so I do believe that they do communicate because they are heavily interconnected. Then the insula is also just one of the criteria for being a hub, but they still, each part performs a different function and each part is just intrinsically quite different. So they are unique, but share some commonalities.
Thank you very much.
Thank you very much, Professor van der Werf. We move on. to Professor van der Heuvel, and she is a psychiatrist, also affiliated to the Department of Psychiatry here at the Amsterdam University Medical Center. Welcome, and the
Thank
floor is
you.
yours.
On the authority of the rector and by virtue of my right, I would like to continue the discussion. But first, really congratulations. It's a beautiful book, and also content-wise, I think it's very, very strong. So well done, also your supervisors. As a psychiatrist, I'm of course very happy that people look at a part of the brain that's not directly or only focusing on motor function, but involving the emotional and cognitive part of the disease. And I think the insula is a perfect spot to investigate interactions between the different aspects. And I think you are a real anatomy neuroscience child, combining pathology, in vivo imaging, linking to clinical practice. So I think, yeah, this is an A&W thesis. So great. I have two problems. So maybe it's not a problem, but maybe you can explain me. The first one concerns the specificity or maybe lack of specificity of your findings. And the other one is the problem of cause versus consequence. So let's start with the first. So very often we compare one disorder with a group of healthy controls and we see something and then we think, okay, what we see is the cause of all things that we see in the clinic. And then you were so clever to also involve other neurodegenerative disorders and see how specific this was for Parkinson or DLB. And then you see, it's not that specific, but you still remain in the neurodegenerative corner of the clinic. And when you then look at other recent papers, for instance, from the Department of Neurology, from Julia Ulegot from last year, he compared, for instance, frontotemporal dementia with many different psychiatric disorders like schizophrenia, bipolar disorder, and autism spectrum disorder. And these people are still young and show exactly the same smaller areas in the insula, the amygdala, and the cingulate cortex. So why do you think this is neurodegeneration related and not just a sign of vulnerability unrelated to neurodegeneration, but in a far broader aspect of brain development, maybe already early on in life or early in adulthood?
I learned upon, thank you for your words. So I indeed, that is correct. And I do not believe that the insular degeneration is neurodegeneration disease specific. Because we know from the literature that it's also involved in diseases like autism and like what you mentioned, schizophrenia, with a study showing that the insula is affected across 26 neurological disorders and heavily affected in these diseases. So we know that the insula overall is a region that is very vulnerable to degeneration. But the reason, for example, we looked at it from a meta-analytic perspective, looking at other neurodegenerative diseases, was that we wanted to understand how it really contributes to other neurodegenerative diseases and what kind of functional deficits would be associated with it, but also where in the insula do we see the same kind of pattern of degeneration as we see in Parkinson's, with more pathology in the anterior and less in the posterior, and what kind of deficits associated with that. So even though I do not believe it's especially neurodegeneration specific.
Yeah, but do you think that the underlying pathology in, for instance, Parkinson's disease or BLD is the same as what we see in schizophrenia or depression? So in fact, when you look at a scan, you do not see a difference what is causing this smaller volume.
Yeah.
But when you go to the pathology, what do you think is the difference explaining the smaller volume?
Yeah, so maybe schizophrenia doesn't have the same pathology, or depression doesn't have, like with other neurodegenerative diseases, but there may be some neurotransmitter deficits in the insula underlying perhaps the damage within the insula. So it may not specifically be related to pathological aggregates, but may be related, for example, from a neurotransmitter perspective.
But when you turn time back 20 years before they develop Parkinson's disease, and you compare their brain with another healthy control who is not developing Parkinson's disease, do you think that there is already a difference in the volume very early on, before real pathology is there due to the Parkinson's disease? Might it be that some people are just more vulnerable?
So indeed, there are studies showing Parkinson's patients in the early phases with mild cognitive impairment, where the insula is affected, showing dopaminergic receptor binding, reduced dopaminergic receptor binding, and that this has to do with cognitive impairment in these particular patients. Whether that particularly has to do with the spread of pathology at the same time, and that is correlated then with the changes in the dopaminergic receptor activity, that I believe is, we don't have an answer for that, because then we're looking at the brain from different levels, different perspectives. But I do believe, based on the literature, that the insula is affected early on, at least from a neurotransmitter perspective. How that exactly has to do with the spread of pathology within the insula, I believe, would need to be investigated.
Mm. And you currently used mostly cross-sectional data. And I was wondering if you, for instance, look at the paper of the Enigma PD consortium on Max Lansma, who's sitting in the room. There you see the different stages with the involvement of all the cortical and subcortical areas. I didn't see a very strong role of the insula in differentiating between these stages. Do you understand that finding?
So that in the cohort there were different stages, and that the insula was not particularly affected?
Not different across the different stages, as far as I could see in the findings.
Yeah, so I think they're just two different worlds, because when we look at it from pathology perspective, we know in Braak stage 5 and 6, the insula is definitely affected, not alone, but also with the cingulate cortex. And that the staging for the different pathologies that are present in Parkinson's, the insula is also affected. But from a macroscopic world, from the connectivity world, why the insula, for example, in a large cohort is not particularly affected, I guess then we would need to look at it also from a perspective of cognition. Maybe if we were to look at a cohort that has Parkinson's with mild cognitive impairment or dementia, that then the insula would show up as being particularly affected.
Is my time over? Okay, then I keep the rest for later.
Sorry, Professor van der Heuvel. Thank you very much for the interaction. Close this sort of opposition with the final opponent, Dr. Lemstra. Sorry, you're a neurologist almost. Neurologist at the Amsterdam University Medical Center and the Alzheimer's Center here in Amsterdam. Welcome, and the floor is yours.
Thank you. On authority of the rector and by virtue of my right, I really like to discuss with you this thesis that is in front of me, but not before congratulating you with this very nice work. I've read it with great interest, and also congratulations to the team that supervised you. You have been traveling to an island, and if I read the general introduction, first, it's almost like reading like a travel guide. Hidden underneath the lateral sulcus lay an island, the insular cortex, also called the insula of Ryle, as it was discovered by Johann Christian Ryle in the early 1800s. And I envision somewhere in the Pacific, a tropical island with palm trees. But if I look at your front, your envision of this island is much more gloomy, is a dark, mysterious island. But nevertheless, you were brave enough to explore, and you did a great job at that. Some of this work is, for me, as just a simple clinician, not easy to understand, but I try to bring this back to my clinical practice. And as you know, I work in the Alzheimer's Center here in Amsterdam, and have a special interest in dementia with Lewy bodies. And there's always this discussion about the relationship between Parkinson's disease on one hand of the spectrum and dementia with Lewy bodies on the other hand of the spectrum. And can you tell me, did you find real pathological functional differences in the insula between these two disease groups? And not only, I saw in your work that you see that especially the load of pathology is much greater in DLB than in PD, but can you maybe tell me if you find other differences as well?
Yeah, learned upon, and thank you for your words. So indeed, we studied dementia with Lewy bodies in the pathology part of the thesis. And we did see that dementia with Lewy body cases had extensive pathology, extensive alpha-synuclein, phosphorylated tau, beta amyloid, CAA, cerebral amyloid angiopathy, also argyrophilic grains. And that seemed to be different from Parkinson's, and indeed quantitatively that also was significantly different from even the Parkinson's dementia groups. And that's also even though there was a huge difference in the disease duration, with most of the time dementia with Lewy body patients having a disease duration of only 3-4 years, and they still had extensive pathology. So we do see the extent of the difference in terms of the load of pathology.
And if you see this in the light of the Braak staging scheme, which actually advocates a kind of caudal to rostral propagation of alpha-synucleinopathy, does dementia with Lewy body fits in this scheme? Because we see already early on clinically many deficits in cognition, but also neuropsychiatric deficits like hallucinations, depression, etc., which you relate maybe to the insular cortex, although I do agree with the opponent next to me that maybe it's not that specific to this disease. But it does raise the question that in this dementia with Lewy body, maybe a different spread or maybe a different speed is going on with alpha-synucleinopathy. What are your thoughts about that?
Yeah, there may be a difference for sure, but I think the main difference what we see in the insula is in the load of pathology.
Mm-hmm.
The load of pathology is extensively way more than any other cases.
But if you look outside the insula and look at the spread of pathology, do you think there's a difference in dementia with Lewy body disease compared to Parkinson's?
So we did not specifically look at other regions neuropathologically, but for example, from the imaging perspective, there are some studies indicating that the dorsal part of the insula could be a biomarker for prodromal DLB because it shows atrophy early on in the disease. And we do see that indeed neuropathologically, that the anterior part of the insula has extensive degeneration. So yeah, I'm not sure about the other parts of the brain exactly, but definitely the anterior insula is damaged in dementia with Lewy body, and particularly the dorsal part of the insula.
And you say, well, even with patients with short disease duration, we see already a high load of pathologies, not only alpha-synuclein, but also a lot of other pathologies. And so my question is, do you think that the DLB fits this Braak staging so that it goes from caudal to rostral, or maybe does the pathology starts already in cortical areas?
Yeah, that is indeed difficult to exactly answer because of the staging and the distribution of the different pathologies, and that they don't exactly happen at the same time in the same regions of the brain. So yeah, whether that fits with the Braak staging, it seems to fit with the Braak staging because we still see more anterior pathology compared to the posterior pathology. But maybe the process itself is different because, for example, we could see also our gyrophilic grains, which we did not find in Parkinson's, and also Parkinson's with dementia. So
there's still a lot to explore, and maybe different islands to discover. But let's stick to the insula because you are very fond of it, and you think also that based on your findings that it could be used in clinical practice as a biomarker. And maybe you can explain how you envision that. How could we use this in clinical practice for as a biomarker?
Yes, so maybe from the imaging perspective, there is, as I mentioned, there's already some data hinting at the dorsal anterior insula being atrophic early on in prodromal DLB. But also from the perspective of understanding a region that is so heavily affected in cognitive control and the way it switches between the large scale networks in order to control cognition. And yet we still do not understand what kind of deficits are present in that particular region and how it could contribute to cognitive impairment. So there, for example, there are a few studies that mention that cholinergic receptors are affected in DLB, and then when patients are treated, that there is improved binding and that is associated with cognitive performance. So maybe if we do understand, for example, the neurotransmitter, the entire neurotransmitter profile of the different parts of the insula and all of the distribution patterns of the pathologies and the methods of degeneration that they induce within the insula, for example, how we have lots of pathologies in DLB and also extensive axonal degeneration, then if we understand it from that perspective, we have more information to be able to say if we do provide these therapies, if we improve the receptor binding or the neurotransmitter profile, that we could improve the functions of the patients. Now that my defense has ended, I sincerely thank the opponents for the attention they have devoted to my thesis. I now look forward to the decision of the doctoral examination committee in the Faculty of Medicine, which will decide whether the degree of Doctor of Philosophy will be granted to me.
And the decision will be made in a closed meeting of the doctoral examination committee. And for this, I adjourn this meeting. Please be seated. I reopen the public session. By its duly accorded authority of the Doctoral Examination Committee of the Faculty of Medicine, grants the degree of Doctor of Philosophy to Yasmine Yousri Fathy. And I invite the supervisors and the co-supervisors now to place their signatures to the degree certificate as witness of this deed. I declare now in public that in the privacy of the closed session, the candidate also placed a signature and by that adhere to the conditions laid down in the provisions in Article 5 of the doctoral regulations of this university regarding rights and responsibilities in society and science. Professor Berendsen is appointed by the College of Deans as supervisor and he will perform the investiture after the signatures have been placed. I now ask colleague Berendsen to resume the ceremony, and during this, only the examination committee stands.
By virtue of the authority vested in me by the statutes of the Vrije Universiteit, and as supervisor appointed thereto by the College of Deans, I hereby confer on you, Yasmine Yousri Fathy, the degree of Doctor of Philosophy, together with all rights, privileges, and responsibilities thereto appertaining. In witness thereof, the seal of the university and the signatures of the rector and the supervisors have been affixed to the diploma you're about to receive.
Dear Yasmine, dear Dr. Yasmine Fathy, I would like to really congratulate you with your endeavor and to really try to understand the role of the INSLA. And I want to congratulate your family too with this great achievement. Like your family is here and even your kids are joining now this ceremony. Chapeau, well done. In the past years, you really showed that you want to understand and learn and with so much passion and devotion what the INSLA is all about. And you have been looking at it from many different perspectives, not only like to really look at it from anatomical point of view, but also from clinical point of view and MRI perspective. So I think this is really remarkable that you can actually put that all together and then still have such a helicopter view to look at what it really means, what we see in this region. You have started in my team in 2014. So it has been quite a long journey, but there was so much to discover. And you were so passionate actually to go into so much details on the structures. And it's not only that we looked under the microscope for many hours to really see what is there, but you also try to train everybody else. I still remember that one time I asked you to give a lecture for one of the students in clinical neuroscience, and you were so passionate so that in the end they would know all the details of the INSLA from the site architecture point of view, from the imaging point of view. So it's really amazing how you brought forward your knowledge also to the next generations. What we also saw during your master's program and also now during your PhD project, that you have excellent writing skills. You love writing. You write blogs right now, and you really want to share your knowledge with anybody around you and also in the world. You've written 100-page master thesis at first, and then said, I really want to stay and learn more about the insula. And I was like, okay, so we have all this information from these different donors, but we don't really understand yet at what stage or which protein aggregates are there, what happens there. And there was so little out there. So you said, I will start with a review to really see what is out there. I think you have brought together like a gallery of images. Like it was mentioned actually that there's beautiful images in the manuscript, but you actually took so much more that you could actually fill almost a whole museum room with it. That was so beautiful and there was so much to discover. And you spent many hours with Professor Rosenmuller actually behind the microscope to really look at all the details. So I'm very proud of you. And I think you did an excellent work with your thesis. And it has really be something that you've been so dedicated to. In the last years of your PhD, you also became a mother. And again, there we saw how passionate you are and how close you are with your family. It was really nice to see your growth through all these years as a scientist, but also as a person. And with so much love and passion for your direct family, but also for your work. So I hope that you will continue to discover the mysteries of the brain and share that with all the people around you and also with the neuroscience field. You've been taking up the task to not only be a neuroscientist, but also finish your medical exams in the next months to be able to practice medicine. And one of your main goals, which you also state in your thesis, is that you want to improve the patient's lives. And that you really want to translate the knowledge that you gain in the research to the patient care situations. So I would like to applaud you for that. And I think you will have a very bright future. You have done so much during these years in your growth. I would like to congratulate you. Thank you very much.
So, to the congratulations of your supervisors, I gladly add also the congratulations of the deans of this university, also, of course, to the family sitting on the front row. You may all be very, very proud of her. I would also congratulate Roche Diagnostics for acquiring you, because I think they have got a huge talent on board now. And I think it's very nice that we work so closely with Roche in this university. So I think we'll meet again, I think, in your next career. I must say I was astonished by everything I learned during this hour about the island. The only island I know in Lewy body dementia is actually the island that you see on FDG PET, where the posterior cingulate is the island, which in the rest of this, there's hypometabolism and the island is lit up. So I've learned a lot. So thank you very much. And I would just echo the words of your supervisors that I hope you have a great career in front of you, and I have no doubt about that. So it's now time to celebrate, and we'll all follow you to the reception, which is just out there. I would ask everybody to stand up. I close this public session of the Doctoral Examination Committee of the Faculty of Medicine. May our search for truth and wisdom be of service to the flourishing of the world and humankind.